Anjali Soni ma'am to kindly introduce him.
Good afternoon, everyone. It's my pleasure to welcome you to this session on skill set requirements in microbiology and molecular biotechnology based industry. Today we have an esteemed scientist who has made significant contributions in this field. We are honored to have with us Dr. Datta Madamwar sir, a distinguished researcher. Right now he is a scientific advisor at PD Patel Institute of Applied Sciences, Charotar University of Science and Technology, Changa, Anand. He obtained his PhD from BITS Pilani. He worked as a postdoctoral fellow at TIFR Mumbai, University of Frankfurt and University of Konstanz and also served at BITS Pilani. Sir has been at the forefront of studies in microbial biotechnologist and his main research focus is on microbial bioremediation, environmental biotechnology, non-aqueous enzymology and cyanobacterial biotechnology. And you know that all these areas are the very demanding areas are nowadays because we are all searching for some alternative to the renewable energy. So these things are very helpful. So he has received lot of honors and awards. Few of them is that Sir is listed in the world ranking of top 2% scientists. He is a recipient of European Commission Visiting Scientist Fellowship. He is also visiting professorship at Swiss Federal Institute of Technology. He is also Lifetime Achievement Award of BRSI for the year 2019. He has published more than 290 research papers and has several book chapters. He has also been the fellow of various organizations like International Bioprocessing Association, fellow of Biotech Research Society of India, and the main thing, he is also working for the social cause which everyone should be aware of, that he has adopted the village in Raigad district for socio-economic development, and in the first phase the school infrastructure of the village was developed and male and female toilets were constructed, and we should appreciate on this that this is a, and sir has also mentioned in the morning, and we should very thankful to you, sir, that if we achieve something and then we go for the society, ultimately everything is to be go to the society. This is so we are privileged to have Dr. Madamwar sir share their insights with us today. Please join us in welcoming Dr. Madamwar sir. We welcome you, sir.
Thank you very much, madam, for such a nice word about me. I just like to point out that I think the last point, which is regarding the toilets and other thing, I think belongs to someone else. It has come to me. I am not involved. I do involve in the social activity, but not in this. There's something else got mixed up. So anyway, I just you should not think too much high about me. That's what I wanted to. Thank you very much, sir. First of all, let me thanks at the outset Dr. Gaurav and Dr. Ambika and team for inviting me to share some of our information with you. I've been asked to talk something about the molecular biology. In fact, as you all know that the molecular biology is considered to be the cornerstone of modern biotechnology. and it has made an impact in various areas. It has been what you call that there has been a lot of innovations and development and which has made a lot of impact in the agriculture, pharmaceuticals, even environment and various other fields also. Also. And there is a demand because it has been having the impact in so many sectors. So there is a demand also with the government institute, government universities or the other private universities, the biotechnology firms, pharmaceuticals firms, and some of the different government programs where these people are required. No doubt when we talk about the molecular biology, we always say that we talk something about the structural and functional aspects of the biomolecules and mainly with reference to the DNA, RNA and protein. Though there are other biomolecules, but these three biomolecules are considered to be the most important. And there is no what you call cellular system available which is divided of these and they have been used for different purposes also. When you talk about the molecular biology, the very first thing in our curriculum, what we try to do that, we try to understand, we talk something about technology, our techniques required for the molecular biotechnology, our microbiology, then we talk something about the application part of that, and the thirdly we talk something about that, how do you perform those experiments in the laboratory. When you talk about the techniques, starting with the DNA, RNA isolation, why do you require to do the DNA isolation? Why do you require to do the RNA isolation? You require that unless and until you isolate the DNA or isolate the RNA, you will not able to understand what are they available, what are their nucleotide, what is their sequence, how they are involved, what are the different types of genes associated with that. So we require to have to a method available by which you try to isolate those molecules and try to. And once you isolate that, whether it is a really DNA or not, what we do, how do we check it? We try to check it either through the initial spectroscopy, and this spectroscopy will give some level of information, but that does not tell you that whether it is really DNA or something else. It will any molecules will give some level of absorption at 260, 280 and so on. So we try to take the ratio to understand the purity. But beyond that, to know about the DNA, we carry out the electrophoresis. Unless and until you perform the electrophoresis, perhaps you will not know that it is really the DNA, and then we try to. So you have to have a technology available by which you carry out the electrophoresis. Once the electrophoresis is carried out, then what we try to do, we try to go for the cloning, recombinant technology, and so on, so on, and then only you can do it. For example, if you have to have a, you want to go for, have to have a particular gene responsible for a particular function. So you try to, you will have that gene, get it isolated, you go for the amplifications with the PCR, qPCR and the thing. And this, because when you get the nucleic acid available from the system, it is available in very, very small quantity. So that perhaps may not serve the purpose for further analysis. You require to increase their quantity and that is possible by the PCR. And if you are interested in understanding their expression profile and other thing, we require to have a quantitative PCR also. You try to understand the number of genes available in the system, then you have to have a qPCR. So you require to learn all those things and then only you can have it. Then you comes to the now gene editing. These days people talk something about the gene editing and as you all know in the year 2020, the Nobel Prize in Chemistry went for the gene editing, that is the CRISPR-Cas9 system. And one of them, that is Jennifer R. Carpentry, she is a MSc microbiology. She was MS microbiology and she did her work in the Max Planck Institute, Germany. And the other were developed. So both were the biologists got the Nobel Prize in chemistry. So you can understand that irrespective of your field, where do you put the applications? That matters a lot. And how significant that application is there, accordingly it will have the impact for these people. Similarly, we also know that The engineers got the Nobel Prize in physiology or the medicines. So at higher level, initially perhaps you have some sort of over specialization, and as you grow at higher level, for example, MSc, PhD, another thing, we talk about the interdisciplinary. And even these days we talk something about the transdisciplinary. And, uh, and in this research world, you require to involve not only the persons of your own field, but you require to involve the persons of some other field also. So you require some sort of a collaboration. You may not be able to do so good work in solo, but your work will carry lot of significance, impact if you collaborate with others. So if you do the exchange of idea, if you get involved with that, then perhaps you can do it much. In fact, in the morning when we had Professor Nitin Desai, to the best of my knowledge, perhaps he is a biochemist by his training, initial training. But is that correct? Biochemist. See the botany. So, and the type of work which he has been doing, because I said biochemist because he talked something about Dr. Sanjay Govinda, who happens to be my friend, and he was in Jipar. So, because he was heading the biochemistry department, that's why I thought that he is biochemist. But the type of work which he's been involved, it's involved in variety of fields. He talks something about plant sciences to the animal sciences, human sciences also, healthcare also. So such a wide area, yet to see the person covering such a large area. You as you grow, you try to have a narrow area. You try to develop a special what you call that you become expert in a specific very narrow area. But he is a person, he at the latter stage, he is trying to widen his area, and that's what is required. That's what is required. You require to learn everything, and then you, after learning everything, you have to have some sort of a all over around knowledge, and then only you can make it a good person, and then you develop the things whatever you want to have it. This is what required. In case of the gene editing, initially it was developed for the plant sciences. People have developed various plant models where certain genes can be silenced, certain genes can be deleted, and see that. And that has been further increased to what is known as metabolic engineering, where you can modify the pathway, and those modified pathways can be subjected for specific product formations. And that has been now further used even in the health sciences, even in the health science, and this is considered like, for example, we talk something about the what you call that you want to silence or you want to remove a particular gene in gene therapy. Specifically in the gene therapy, it can be used. So like that in every field it has been getting expanded, and that's what happening in this field. So there are a lot of like that. You have to have a knowledge something about the what is known as the bioinformatics and data science. Why do you require? Once, for example, if I am interested understanding the diversity of organisms available in a particular system, what I would do? I would try to get the total DNA from the environment. Once I get the total environment from the environment, I will go for the whether it is a, if I am interested with the microbial bacterial system, I will go for the 16S amplifications. And after that, I will go for the sequencing that 16S and I get the information about the only the bacterial system. But if I am interested about the fungal system or other type of microorganisms, I require the ITS results also. So I will do that amplification, I will do the sequencing. And once you have those sequences available, how do you process those sequences? You can process those sequences provided you use certain platform. Unless and until you use certain platform, those sequences will not be utilized. And for that you require data. So you have to run several whatever softwares which are available, and then you have your data, put those data, try to see, align this data and try to get the information. And then only you will come to know that what type of organisms available in that. Similarly, if I am interested in understanding the functional aspects of those, for example, if I consider a particular ecosystem, if that ecosystem, what type of organisms are available, how these organisms are interacting with each other, what are they doing, how do they act, for example, if that ecosystem is having some contaminant and those contaminants are getting degraded, those contaminants are getting decomposed by these organisms, how do they do it? So if I am interested in understanding about that, I will try to understand the functional aspects of that. And to have that, you may get some information using latex, for example, Priscus, which will help you to have a predictive functionality. Predictive in the sense that you know that this type of organisms are available in that environment. Those organisms will let you know what type of those organisms, what are their genes available, their data banks are already available. So based on that, you can understand that these organisms are involved in this process. So that is on prediction. But if you are really interested in knowing that really are these functions are being carried out or not, so what we do? We have to go for the what is known as shotgun metagenome sequencing. And when you do the shotgun metagenomic whole metagenome sequencing, then you will have lot of information, different types of the genes which are available and what are those genes doing, and then you can have a cake pathways and other things. So you will come to know different types of pathways which are being operated. So you can understand that how these organisms are associated with each other and thereby you can get the information. Unless and until you have that. So this is what happening. Similar thing is happening even in the pharma sector. Similar thing is happening in the biotech sector. So in all the cases, you require to have these information then only. And many times what happens that simply by, for example, if I let us say take the DNA extracted from a particular ecosystem, I get the type of microorganism available based on the 16S amplifications. I carry out the shotgun metagenome sequencing, I come to know the different types of the genes. What is happening that there are certain dead organisms available. If there are dead organisms, their genes also will come in the picture. So to avoid that, what we can do, instead of going for the DNA extraction, I will go for the RNA extraction. RNAs will be produced only by those organisms which are alive, which are involved in the various process. Otherwise, they will not. So you take out the RNA, and if you have a RNA, you have to have a cDNA preparation. So from the RNA, you will go for the cDNA preparation. If you do the cDNA preparation, you will have go for the sequencing of those cDNA. So lot of then you can have some. So you are having what is known as metagenomic. You have a process what is known as metatranscriptomics. So that is about the RNA's information. Even many times this will not serve the purpose. This will still not serve the purpose. What we do that? We go for the metabolomics. We take out the whole samples, take out the extract of that sample. After taking out the extract of the samples, there are processes what is known as solid phase extraction or liquid phase extraction. Take out those samples and again subject it to the what you call HRLCMS. So if with the HRLCMS, that is a high-end LCMS, there you will know the different types of metabolites which are available in that. So you will have. Similarly, we also try to go for the total protein extractions, and after getting the total protein extraction, they are also subjected for the MyData. Again the LC. But that is exclusively for the protein. So you have a what you call the metagenomics, you have metatranscriptomics, you have metabolomics, you have metaproteomics. So you have all omics technologies and they are all integrated together. So you can have integrated omics technology. And there what you require to have a role of what is known as artificial intelligence and machine learning. If you cooperate artificial intelligence and the machine learning in the multi-omics technology, you will have. So for that, you require a computational scale. Unless and until you have that computational skills, you know what type of the platforms which are available, utilize those platforms, analyze the data, you require a very so you require a bioinformatics also, the data processing. Unless and until you process, you get lot of data, but this data has to be further analyzed. Unless and until you analyze this data, perhaps it becomes difficult to understand. And because of the development of these technologies, things have changed. In the next another maybe decade or couple of decades, in fact, he said in the morning also that we are going to, now we are in the what is called the developing country. By 2047, it is targeted that it will be developed countries. And when you have to have a developed countries, what is going to be our goal? So you are, and we all know that, for example, I talk because I come from the environmental background. One of the problems which we face is about the microplastics. Plastics which are being disposed of. And we also, all of us know that finally these plastic finds a way into the waterways, including the sea. And then these plastics are also found in the fishes, aquatic animals. These are found in the various other animals also. So, and even we did, for example, we use the paper cup for tea or water. Even that paper cup, though it is for only layman's sake, they said it is a paper cup. But if you see the inner layer, it has a microplastic. So I don't say that plastic is obviously is a good material. You cannot avoid, you cannot survive without plastic. But it has to be used with caution. It has to be used judiciously. It has to be used in specific ways. Then only perhaps it will work. Otherwise, you are going to face a lot of complications. But then, whatever the plastic which has gone into the environment, you require to degrade them. Are you required to utilize them, recycle them for some other purpose? And people are doing, lot of people are working on the reutilization of the plastic. But the things which are already done, you require to remove those plastics from the environment. And for that, you require organ. And here what again it helps you by carrying out the metabolic engineering in the microorganism. Perhaps you will be able to develop a strain, you will be able to develop a group of organisms, not necessarily one organism, because single organism very rarely works well. But if you have a, like, as I said that, in collaboration, our output will be much better than in solo. Same is the case in the environment. Alone organism perhaps may not do that good job, but when they are in a group together, when they are in the consortium form, they may be in a better form and they will be able to utilize this compound in faster rate and able to convert them into the CO2. Or they will be able to, what you call, so you can have it. Now these days, there is a what you call the fights or you can say struggle which goes between the chemistry or chemical engineers and biochemical engineers. The way chemical process are being produced and to get a particular product are the various chemical process are being carried out to generate certain materials. Now even biological process are also being carried out to generate the similar. So you can have it like that, what I said, the plastic, you can have a degradable plastic. You can have a composite plasmid which is easily degradable and which can be not only by chemical method you can produce, but you can also produce by alternative with the biological system. So thereby you can manage these. So what I mean to say that whether you are a microbiologist or a biotechnologist or any other life science students, there is a heavy demand in the market. You are required for variety of processes. You are required maybe in the government institute, academic institute. You are required in the pharma industries. You are required in the what is called agro industries. You are required in the biotech firms. and you are also required in the environmental programs. So there is a need for these people, but as I rightly pointed earlier, also said that unfortunately we are producing the people much more than the required one. Even in the engineering science also, engineering points also, you can see that the demand for particular group type of engineers, for example, these days there is a heavy demand for the computer engineers and IT people. And their engineers, they are absorbed in the industries in thousands. They are not absorbed one or two. Whereas biotech person is absorbed in the industries in countable number. They are not taken in hundreds. So obviously the requirement of these people is not as high as what you find in the engineers. So therefore our curriculum, our number of people which to be produced by the academic institute should be manageable properly as per the requirement. If you have that ratio properly, perhaps things will work very well. And of course, people, the present, what you call the national education policy, promote to have more of startup, promote to have now there are so many incubators which are available. There are Section 8 companies which almost every university has. Every university has a startup cell. Every university has got a incubator cells. And on the top of that, there are certain government agencies. And Gujarat perhaps is leading in this respect. I'll tell you the very small thing. We have what is known as Gujarat State Biotechnology Mission. If at the central level, there is called Department of Biotechnology, which is an outshoot of the Department of Science and Technology. Now Department of Science and Technology is also been getting renamed. It is getting in different way. It is getting dissolved and it is, that's called that Anusandhan mission. Okay. So there is a, but Department of Biotechnology came into the existence to promote biotechnology. And they started giving the research projects to different, what you call the investigators. And it started like that. Meanwhile, they also came up with their own institute. The very first institute came as a part of the Department of Biotechnology, that is called the NII, National Institute of Immunology, New Delhi. And later on now we have many such institutes like NCCS Pune, FISHI, Regional Biotechnology Center. There are many institutes now, like the Center for the DNA fingerprinting. So we have many institutes which are part of the Department of Biotechnology, and Department of Biotechnology provides the grants to the different investigators. Looking at that, Dr. Manju Sharma was one of the first founder, director, or secretary of the DBT was the Dr. Ramchandra, but the second one was the Dr. Manju Sharma. And Dr. Manju Sharma was the principal scientific advisor to then Chief Minister Dr. Narendra Modi was the to them, and they formed the what is known as Gujarat Council of Biotechnology, and that there she was heading that council. The chairman was the chief minister, but then she was the advisor, main advisor, and that's all. And they came out with the like DBT. Gujarat should also have some center which can allow the younger faculty to initiate biotechnology research, even different colleges and other things. And initially, of course, they were giving small grant. Department of Biotechnology in New Delhi also started having a what is known as capacity building program, which is run even at the GSBTM here also. In the capacity building program, what they do it, in that after the completion of your BSc or after the there are capacity building two types of program. One is called that, there you get a what you call crash program of for clearing the some sort of a national level examination, whether it is a UGC NET or something like that, CAT or yeah, GATE, GRF. But in addition to that, they also started a program of having a training to this student in the industries. But being a department of biotechnology, because they were having fairly good grant with them, they were giving the support to the student in the form of stipend. In addition to that, they were also paying the money to the industry. It is not only that. So, the government of India was spending per student maybe about 20,000 or 30,000 per month. And you are giving training maybe for a period of 6 months or 1 year or something like that. That is after the completion of MSc. And then they were saying that if the candidate is good, the candidate will be absorbed by that. Similar program started by the GSBTM. There also they were not only providing the grant to the stipend to the student, they were also paying to the industries. So they could identify several industries. But the number of industries which are available, which are biotech based or pharma based, in pharma based again there is a competition. Why they will take only biotech student or microbiology student or life science student? They will take the pharmacy student also. But to encourage that, even the government of Gujarat, the first state in the Gujarat, first state, not first state in the first state in India, came out with this suggestion and they started this program where they provided the stipend and every year now it is been taken care. After the completion of BSc, after the completion of MSc, you can have 6 months or 1 year program in that. And where you get a stipend and even the industry is also paid for that. But when you have to integrate similar program during your MSc, where there is no grant available to support to the student in the form of stipend, nor there is a grant available to support to the industries, we do have a problem. It's not going to be that easy. It is only on the personal request, as Dr. Joshi said, if you interact with him and if you request him, he may take. But that doesn't mean, but when you have to have the university set up, you require to have accommodations. Let us say you have 75 or 60 students of your MSc biotechnology, they have to be accommodated in the industries. And fortunately, Gujarat, specifically this belt, Surat, has good number of diagnostic companies here, good number of other industries also here, which are not directly related to the biotech, but still biology related companies you have here. But it is not true. There are very few companies like something like a Zydus, something like Cadila, Intas, Sun Pharma. They have a fairly good R&D centers. And they can absorb only few students. They cannot absorb large number of students. Then what happens that people will try to go, there are certain, what you call that, companies which have come up and they perform they do the R&D work or something like that and they carry out the project work on payment basis. And what do? We all know that very well. So here, unless and until you have a proper defined structure to have training, it becomes extremely difficult and we are going to face that challenge in next few years, how to accommodate that. Somewhere something has to be and it has to have a proper policy. Unless and until you make a proper policy, it will be quite tough. So, but then maybe government once get involved, they will do something or other thing. But microbiology, molecular biology, they are the what you call that heart of any life science, heart of biotechnology. Without that, which will have a lot of difficulty. Without that, it is difficult to learn. So what I meant to say that it is one of the most interesting area where one can get involved and then you can. And to the Dr. Joshi, he said that because you require to think about whether you are interested in R&D, whether you are interested in the production, whether you are interested in the regulatory affair, whether you are interested in the particular area. Obviously, even at the MSc level, it becomes extremely difficult to know that which particular portion of industry is more interesting to us. Majority of the students think that R&D is the best. And one of the reasons why R&D is the best, because here you have a scope of learning more and more. In all other, whether it's a regulatory affair, whether it's a production, or whether it's a quality control, here you have a monotony. Maybe first year you try to learn, second year you develop the expertise, but then third year onward you start getting bored in that. That's the reason. And that's why we want. But in R&D perhaps, because every time you try to have something new in that, and therefore people are more interested. And this is the jargon basically. People are more interested. But then others are equally. For example, production is equally important. If you do small modifications, small change, and the production capacity can go up, which will earn a lot of money, in that case, that's what happens. So anyway, I try to convey some of my informations to you, and we'll have then maybe some more question answers once we. Thank you very much. Once again, thanks for.
Thank you so much, sir, for your invaluable insights. Our next key speaker is Mrs. Parini Kapadia, ma'am. To introduce her, I invite Dr. Ruchi Desai, ma'am, to welcome.
Good afternoon, everyone. Today, I have the privilege to introduce one of our former students, Mrs. Parini Kapadia, founder and chief scientific officer, Zero Cow Factory, Surat, India. She has secured her master's degree in biotechnology, Department of Biotechnology, in South Gujarat University. She has also completed postgraduate diploma in advanced clinical research and pharmacovigilance Institute of Clinical Research, India, Mumbai. Her work experience shares. She is the founder of Zero Cow Factory, Surat, India, which develops a technology to reproduce milk proteins to craft real dairy products that are safe, delicious, and identical to cow milk without any animal involved. She is part of Czech Incorporation part-time, which provide the technical assistance to the global users for biology and related subjects. She is also involved at Anand Microbiology Laboratory, Surat, which process different clinical samples, identify pathogens, antibiotic sensitivity testing, etc. She is also associated with Perfect Diagnostic Private Limited, Surat, which process blood samples using BACT Alert 3D60, hospital surveillance work, and preparing antibiogram. She is involved at Zydus Research and Development Center, Ahmedabad. Her area of interest are microbiology, biotechnology, clinical research, pharmacovigilance, and entrepreneurship. I welcome her to share her experience. Welcome, Pai.
So, hi everyone. I am Parini Kapadia. I am a co-founder and chief scientific officer of Zero Cow Factory. So, first of all, I would like to thank Gaurav sir and whole biotechnology department to invite me and uh, uh, to give me the privilege to share my journey and what is my experience to share about Zero Cow Factory, what I have learned from that particular journey. So before going to the that particular, what are the skill sets which is required for actual industry, what I have learned, I would like to share about the what is the Zero Cow Factory. So basically, Zero Cow Factory is a precision fermentation based company. So we are producing the dairy proteins without any animal. So we are using two biotechnology concepts. Those are the like strain engineering, that is the bioengineering the microbes, and precision fermentation technology, that is the part of the molecular biology. So Sir has said very well that microbiology and molecular biology is like a heart of the life science subject. So yes, so whenever you have seen any kind of the area, let's say for example pharmaceutical lelo, yeah, any kind of the food industry, dairy, CPG, everywhere microbiology and molecular biology is a core skill set or the core area which is involved. But here I would like to give you the example ki what are the skill set from my journey, what I am doing currently. Let's say for example, molecular biology means it's a very vast subject, right? It can involve gene therapy also, which is another. Then molecular biology that is required for the pharmaceutical products also, like to develop any kind of the drug molecules. There also molecular biology is required. Another industry is like food, dairy, CPG, any kind of the enzyme production, any kind of the protein production. There also the molecular biology is required. So here I would like to explain about the this particular industry like the enzyme or protein production, where which kind of the skill set is required for the in the field of molecular biology and microbiology. So let's say for example, if you would like to go for the any kind of the protein expression or any kind of the enzyme or any kind of the molecule expression, you require different kind of the skill sets. I can say the big revolution in the field of the molecular biology, that is the insulin production. Second, it's the rennet. So you know that till 1990, whatever the cheese we are eating, that is the non-vegetarian. So for the cheese production, one kind of the enzyme is required that is called as a rennet, chymosin. Okay, so before 1990, that enzyme is isolated from the fourth stomach of the baby cow, right? And that is not a sustainable solution. So for evolution and for big revolution is that using microbiology technology, using recombinant DNA technology, people have produced that particular enzyme using microorganism. So now from this example, you can consider how important is the molecular biology for the revolutionary generation, right? So here also in Zero Cow Factory also we are also trying to replicate that particular idea, because whatever the proteins or milk proteins we are producing, because animal agriculture is not a sustainable solution, right? Whatever we are eating, milk, protein, anything, that is not a sustainable solution. There are many disadvantages or many problems are associated with the animal agriculture. Animal agriculture is like a highest contributor for the greenhouse gas emission. Data has predicted that it is equal to the transportation industry. Even it's a higher than the transportation industry for the greenhouse gas emission. You require huge amount of land, water, resources and everything, right? And milk, why we are consuming milk? For protein, right? But you know, to get that particular protein, we need to feed the 6x amount of the protein to the animal, right? And in reverse, we are getting only 3x amount of protein. So that is not a sustainable solution. Okay, that ratio should ideally reverse. Okay, so there are many disadvantages, lactose intolerance. Okay, in our like nowadays people are now aware about the lactose intolerance and everything. Okay, but earlier people doesn't know about that particular thing. So due to this, all the problems, we have started this particular company. They're called Zero Cow Factory, and it's actually the area of cellular agriculture. Now, what is the cellular agriculture? The cellular agriculture is that key production of the any kind of the molecule without any animal. So let's example, without any animal, any kind of the product that involves eggs also, that involves chicken also, that involves meat also, right? And in that there are two particular, in cellular agriculture there are two particular division. First is the cultivated base. So you know the stem cell, animal biotechnology, that is the one particular division involved into the cellular agriculture. And second is the precision fermentation, where the core microbiology and this fermentation technology are involved, right? So this is the very new innovation, cellular agriculture. In India is a very nascent subject, but outside the India, there are big innovations are happening. There are companies are available, and in India, the products are also available. You can get the milk without animal, chicken without chicken, egg without egg. So for egg production, we don't require hen. Okay, but they are replicating the same product and everything. So that's another part, but let's focus for the protein production. How am I how we are producing that protein, and what are the skill set required in that particular area? So what I can say, first of all, if we would like to produce any kind of the molecule, the journey first of all start with the subject called as a bioinformatics. Bioinformatics is very important because to understand the protein or any kind of the product behavior. Let's say I'm going to produce the milk protein, but bioinformatics help us to identify the particular fragment or DNA sequence that is responsible for the production in cow, right? So that particular identification, bioinformatics help us. So bioinformatics is required in that particular area. So we can use bioinformatics tool, we can identify that particular fragment or gene in which particular chromosome it is present and everything, right? And before starting any work, if you would like to start any kind of the protein molecule, it is also important to understand the protein chemistry, means how that particular protein, how that particular molecule is there. For that also, bioinformatics tools are very important. So whenever you think about that, the basic knowledge about bioinformatics is very important. If you would like to start like for gene therapy, also gene editing, but for that also bioinformatics is very important. So for that you require the basic understanding, basic kind of the tools or software which is used for the bioinformatics. Second step, I would like to say it's like a molecular core, molecular biology, recombinant DNA technology. So once we have that particular gene fragment identified and everything, we can synthesize those particular fragment in laboratory environment, right? And then whenever we think we will replace that particular thing with the microorganism, now the host selection, that is, there are variety of the microflora, like microflora is a huge, plants भी आ जाते हैं उसमें, bacterial भी आ जाते हैं, yeast, everything. Fungal systems are also nowadays the evolutionary host organism to production of any kind of the product, right? So host selection is also very important. वहाँ पे like the different knowledge regarding that particular host selection. Okay, people in industry, what kind of the host are they are using for such kind of the innovation in bacteria, in yeast, in fungal. Let's say for example, whenever we think about this particular kind of the area, bacteria में Escherichia coli are most important. Bacillus species are they are like famous for that. In yeast, to like Saccharomyces cerevisiae, Pichia pastoris, even the fungal system. Nowadays, people are using fungal system also to produce different kind of the enzyme. For example, Trichoderma raceyi hai, Mycelioptera hai. These are the novel organism, right? So host selection or host strain selection is also very important. And that is also depend on to the molecule, means what kind of the molecule, what kind of the enzyme you are going to produce. If it is a small molecule, then you also need to understand that molecule is able to produce in this particular microorganism or not, right? The host strain selection, that is also very important parameter. For that also, you need to study about that particular system. Let's say for example, I am going to use the yeast. तो वो yeast के बारे में जानना means to understand that whole yeast system is also important. What is their metabolic pathway? What is their metabolic engineering? How my protein are going to produce? That all kind of the information is also required. And then after host selection, the plasmid and everything, that is the general thing that we can learn. Okay. Once the recombinant DNA technology happen, like we have the modified organism and everything, then we'll go for the bioprocess technology. So bioprocess technology, we can't say it's a pure molecular biology, but whenever you choose molecular biology, the basic knowledge about bioprocess technology is also important, because whenever you are going to produce any kind of the organism, bioprocess is also important. Means, for example, my target is to produce any kind of the molecule. For that, how that particular protein is going to produce, what kind of the media, what kind of the fermentation technology we can use. So that is also important. So bioprocess technology, that is also important. Means in that, two particular subtypes are there. That is, first is USP, upstream process development, and downstream process development. The upstream process development that majorly cover the all kind of the optimization in fermentation, and downstream may all kind of the purification, and if whether some kind of the modification is required for that functionality or not. So I can say ki that kind of the knowledge is also important if for apart from the core molecular biology subject, because if you will produce the clone, but if you don't have the knowledge of bioprocess technology, then how you can prove whether you have produced that particular clone or modified organism that is correct or not, right? So that is also important to have this some kind of the basic, not in depth or not like an expert, but some kind of the knowledge ki this particular organism, this screening and expression, everything you would like to know. And after that, formulation, little bit, not like ki you can be expert of formulation, but ultimately this is the whole cycle, that is, these all the area are interconnected. Then you have the one product in the market. Like I am going to produce the milk protein, so I need to understand about the bioinformatics also, molecular biology also, bioprocess technology also, DSP also, and ultimately I need to understand about the formulation also. Then I am able to, or I am successful to launch my product in the market, right? So though if you have the one area of interest, you are the expert in that particular area, but ultimately you need to try to understand about the, or it is good if you have the basic knowledge about the other interconnected area, that will help you to grow in your career. So these are the techniques that I can say, and what I have learned from my journey from Zero Cow Factory. These are the area you can gain the knowledge, and I can say ki don't depend on this bookish knowledge. Just update yourself with the industry also, right? What is going on new in the, because CRISPR-Cas9 and everything is a very new technology, right? In CRISPR-Cas9 also, every day or every time there are new revolution are happening. Okay, first of all, whenever we say CRISPR-Cas9, so that is limited to the plant only, right? But nowadays people are using for the fungal system also and yeast system also. That is the very new innovation, right? So update yourself with the industry also, okay, what is going on in industry, right? So ultimately that will help you in career. Okay, so thank you so much.
Thank you so much, ma'am, for sharing your views with us. Our third key speaker is Mr. Alok Vaidya, sir. To introduce him, I invite Ms. Upeksha Patel.
Good afternoon, everyone. It is an absolute honor and privilege to introduce our next keynote speaker, a visionary, a pioneer, and a true leader in the field of biotechnology and molecular research, Mr. Alok Vaidya, sir. Sir is the director of SLS Research Private Limited, Surat, a company dedicated to cutting-edge advancements in molecular diagnostics and biotechnology. His academic journey is equally remarkable. He holds a master's degree in applied genetics from Bangalore University and another in genetic epidemiology from the prestigious University of Sheffield, United Kingdom. His academic background has equipped with his deep expertise in microbiology, biotechnology, and molecular biology. His professional journey reflects a strong commitment to scientific excellence and industrial research. As a leading force at SLS Research, Sir has played an important role in molecular diagnostic development. Sir played a crucial role at Saffron Life Sciences, Gandhinagar, where he is contributed to research and development in diagnostics. Sir also has extensive experience as a junior research fellow at Navsari Agricultural University, where he worked on applied biotechnology projects funded by the Department of Biotechnology, Government of India. Today, Shar will be sharing his ideas on skill set requirements in microbiology and molecular biotechnology based industries. Please join me in welcoming Mr. Alok Vaidya sir.
Thank you, ma'am. Good afternoon, everyone. First of all, I would like to thank Gaurav sir for inviting me and giving me this opportunity to, share a few things with you guys. अभी इतने बड़े बड़े लोगों ने इतना कुछ बोल दिया है, मेरे लिए कुछ बचा ही नहीं है. So, anyways, so I'm here to talk about the challenge that we are facing right now, like aligning the skill set with the requirement of the industry, right? So इस फील्ड में जो सबसे पहला स्किल रिक्वायर्ड है, आई थिंक दैट इज पेशेंस, right? Because it's an ever evolving field and kindly a slow starter, right? You cannot compare yourself with a computer engineer and expect 40,000 rupees for your starter job, right? So don't compare yourself, be patient. First objective for your job should be learning new technology, new things, rather than to earn money, right? Money will come, you just have to be patient, right? You have to provide your value to the company, then only you'll get the raise. Yeah. Next skill I would say is the mindset of lifelong learning. That is another major skill required in this field, because my mentor used to say, whatever you are studying in your classroom right now will become irrelevant when you pass out and go to the industry, right? So it's an evolving field, right? Rapid evolution is happening in every day, right? New innovations are coming, new technologies are coming. So you have to keep yourself updated, know what is happening in the industry, know what kind of technologies are coming up, then only you'll be able to get the desired job. And next, I think, is the basics of microbiology and molecular biology. Because you are in this field, when you go to industry is not expecting you to, be an expert in anything, but at least the basic fundamentals of the field, molecular biology and microbiology, that industry expect that the freshers should know. The techniques like DNA extraction, RNA extraction, PCR, right? Plasmid extraction, basic bioinformatic knowledge. These are the basic things that industry expect that from a fresher after MSc. Right, so that's the technical skills that I am talking about. Next is another major important skill is soft skills, because टेक्निकल स्किल्स के साथ साथ ये होना भी बहुत ज़रूरी है. हाउ यू अप्रोच अ कंपनी, राइट? कोल्ड कॉलिंग, कोल्ड ईमेलिंग, ये भी आपको आना चाहिए. कम्युनिकेशन स्किल्स होनी चाहिए आपके पास. यू शुड बी रेडी टू कोलैबोरेट, बिकॉज विदाउट कोलैबोरेशन कुछ पॉसिबल नहीं होगा इस फील्ड में, राइट? तो जो सिक्रेटिविटी, जो सीक्रेसी जो रखते हैं एकेडमिक स्पेशली, ये मैंने देखा है. मैं भी ऐसे ही था पहले, जब मैं एमएससी या प्रोजेक्ट में काम कर रहा था, तो जो प्रोजेक्ट पे काम कर रहा है किसी का शेयर नहीं करते. ऐसा लगता है कि हम कुछ बहुत बड़ा कर रहे हैं, किसी को पता चल जाएगा, कोई चुरा लेगा. राइट? तो ये नहीं है, आप जो सोच रहे हो, जो कर रहे हो, वो ऑलरेडी किसी ने कहीं ना कहीं कर लिया है, राइट? सो वॉट एवर यू आर वर्किंग ऑन, जस्ट सिंपल, अभी अभी आपको कोई आइडिया आ रहा है, एक बार उसको सिंपल गूगल करके देख लेना, एआई वेआई छोड़ दो, सिर्फ गूगल में सर्च करके देखना, किसी ना किसी ने कहीं ना कहीं पे ये कर लिया होगा, राइट? एंड again, one of my mentor used to tell me that never waste your time, energy and resources in reinventing anything, राइट? जो इन्वेंट हो चुका है आप उसको यूटिलाइज कीजिए, आप उसको इम्प्रूव करिए, उसको फिर से री इन्वेंट करके कोई फायदा नहीं है, कोई मतलब नहीं है, राइट? सो कीप एन ओपन माइंड, बी रेडी टू कोलैबोरेट विद अदर्स, बिकॉज ये इतना बड़ा फील्ड है कि आप अकेले सब कुछ नहीं कर सकते, राइट? अगर mRNA वैक्सीन बनी है कोविड का इतना जल्दी, सो इट वाज बिकॉज अ टीम वाज वर्किंग, राइट? बायोइन्फॉर्मेटिशियंस थे उसमें, राइट? वैक्सीन के लिए जो जो चीज चाहिए वो हर कोई एक बंदा नहीं कर सकता, राइट? सो कोलैबोरेशन इज अ मस्ट, सो कीप एन ओपन माइंड, yeah. And I think another skill required in this field, yeah, soft skill, as I said, approach आपको रखना चाहिए, and lifelong learning is the most important thing. Uh, yeah, these are the basic skills for you getting your first job in the industry, right? So बहुत basic सी चीजें हैं जो आपको इंडस्ट्री में एंट्री दिला सकती है, राइट? देन इट इज अप टू यू कि आप उस इंडस्ट्री को क्या वैल्यू प्रोवाइड करते हो, दैट विल डिसाइड योर जर्नी अहेड, राइट? सो होपफुली every one of us will work together to create an ecosystem where talent can thrive, innovations can flourish, and India hopefully will lead the biotech revolution in global stage. Thank you.
Thank you so much, sir, for sharing your views with us. Now we are opening the floor for question and answers. Feel free to engage in discussions.
So my question is, life science graduate opportunity is very less compared to foreign countries, and space skill is also very less. So what measures can be done at institute level or university level to compete it?
I think it is according to the economy of country rather than anything else. So, if you are good, it doesn't make any difference whether you are from life science, whether you are a microbiologist, or you are a botanist or geologist or whatever it may be. I think at higher level, it hardly matters. What is more important is that if you are good and have a, what you call that, aptitude of working or learning, you should not have any problem. Initially, obviously, you may not get good scale. He rightly pointed out, don't worry about the initial. You consider initial job as a sort of a learning experience. Once you learn and you develop an expertise, then you will have a lot of scope of improvement, getting the good salary. Should not be a problem. So,
Yeah, sir is absolutely right. ऐसा नहीं है कि जॉब्स नहीं है लाइफ साइंस में, लाइफ साइंस में फ्रेशर्स के लिए कम जॉब है, because it's a very specific field, right? And the equipments and the materials and the process involved is highly sophisticated, and a lot of money is involved for the industry. So freshers को पे स्केल इतना ज़्यादा नहीं मिलता, because company is not expecting a lot of work from the freshers, right? Until and unless the major objective for the student should be to get in the company of their choice of field. And once they get in, they should learn and prove themselves. Then definitely they'll get the rise. And there are many life science students who are earning way more than computer engineers and other professionals.
We are here talking about skills and acquiring skills and skills enhancement as per the need. Many of the students are there, just I want to ask them, how many of them come by a bike? You back in love with your bike, right? You are in love with your bike, all of you are. You know each and every part of it, how much oil, how many colors, which make, uh, which year it has been manufactured, what modifications, all what they have done. Just want to ask, how many of you know what is laminate made up of your lab? Which company manufactured? USC? Any like local manufacturing company which manufacture pipettes? अंकल अंग्रेज अपने इक्विपमेंट को गले नहीं लगाओगे स्किन नहीं आएगा फिर आपकी डिफरेंट नहीं होगा you have to see the equipment thoroughly, understand it, understand its utilization, understand its troubleshooting also. That is what industry wants. Industry को ऐसा जॉब चाहिए जो सब कुछ काम करे इसलिए right? That is very important. So whatever you learn in theory, go to the lab, learn everything at once, your own skills, adapt new things. That is very important. Now, somebody asked me salary. Yes, you can adapt to hourly basis salary. What is happening in US? Hourly basis contract. You are good enough, come and do this work and go. The skills going beyond, like for example, the skills of a carpenter, a plumber, a driver. I'll give you one example. My driver gets a 24,000 salary. My own nephew, ME mechanical, is 14,000 salary. My driver is illiterate completely. He has patience. He drives for 12 hours, he works with 24 hours. Whereas my nephew was only for 8 hours and it's be mechanic. 14,000 salary. But at certain times you have to have patience initially for skilling yourself, updating your knowledge. These are initial investments of your time, initial period. From day one, you cannot take sitting in the AC and signing the checks and expecting salary 1.5 lakhs. So we all need to invest some time. Everything comes with age and experience. That's why experience matters. Right? Like for a teaching job, we enter as assistant, maybe I, as assistant, then associate, then professor, senior professor, and so on. Same is the case with IAS officer who enters as the IAS. He was same salary like a lecturer, basically same. Because secretary, chief secretary, he was on the verge of retirement as a senior. So same is the case with the industry. But in the industry, people work round the clock. They are always busy, they are always on the go, they are always product. So you just compare whether we are that much productive or not. And there what I feel biologists exclusively, you get carried away. And they do not focus much on commercial part. And that's the reason there are less opportunities that you see in biology. But days to come, biology is going to take over most of the industry. When we talk about polypeptide life, food, nutrition, agriculture, there are a lot of things that will come. We need to adopt this new coming technology. Do new additional courses. There are a lot of ample time, a lot of universities are there online, giving so many teaching classes, technology. have done great job during your COVID course itself. All the classes you have come will have done. That is remarkable. I'm very happy that your students are coming and sharing their knowledge. They are already very professional and successful students. So if you let them copy them, there is no harm copying somebody. Something is good for you. Right? Thank you.
Thank you so much. It was indeed an inspiring session. As we move forward, I would like to request Dr. Mayuri Rathod ma'am to felicitate Dr. Datta Madambar sir as a token of respect and gratitude. Ma'am to felicitate Mrs. Parini Kapadia ma'am. Lastly, I request Dr. Gayatri Solanki ma'am to felicitate Mr. Alok Sir as a token of appreciation. Thank you so much, everyone. Let's take a short tea break before we return for an engaging panel discussion. We'll get